Greeting to Dr. Haru. The title of our assignment is Molecular Mechanism of Gene Expression in Human Cell. Group members include Hyun which is me myself, Hugh Patient, Usna Fariha Binti Hasri, and Chong Sun Seng. Now I will pass the presentation to my groupmate Patient.
Technological advancements now enable investigation of molecular profile changes such as mRNA, protein, and metabolite levels, which can aid in understanding the complexity of the gene disorders. Gene expression provides comprehensive information on the genome location, pathway, and annotation. It can be viewed as a refined phenotype, a viable method for identifying the qualitative trait loci, known as the QTLs, and comprehending the genes, pathway, and biological processes affected by these loci is by integrating this with the genetic inheritance data. Human cell gene expression is induced by the interferon that triggers the initiation of the transcription, which result in the synthesis of different proteins. Next, we are going to talk about the molecular mechanism in regulating the human cells.
Transcriptional regulation is a critical process which enables the cells to respond to signal to, to maintain their identity and coordinate activity. An example of transcriptional regulation is hypoxia and hypoxia-inducible factor 1 (HIF1). Under hypoxic condition, HIF1 alpha stabilize coupled with HIF1 beta, and this complex bind to hypoxia-responsive element. This will induce the transcription of genes involved in angiogenesis, erythropoiesis, and metabolic shift. In cancer, tumors often overexpress HIF1. This will promote tumor proliferation, survival, angiogenesis, and metabolic adaptation. Targeting HIF1 is a promising approach in cancer therapy. Strategies involve small molecule inhibitors, gene therapy, and immunotherapy with vaccines or antibodies. Moving to RNA processing, which is critical for transition from gene to protein. An example of RNA processing is SMA2 gene in spinal muscular atrophy (SMA). The severity of SMA is influenced by splicing of exon 7. This is because the presence or exon of this exon will affect the protein functionality essential for motor neuron survival. Therapeutic strategies have been developed using antisense oligosaccharide to promote the inclusion of exon 7 in SMA2 mRNA. This will generate functional protein to alleviate the symptoms. Now, I will pass the presentation to my groupmate Chong Sun Seng. Thank you.
Next is translational regulation, the process of regulating the synthesis of protein from mRNA templates. Ion response element (IRE) system is an example of translation regulation. IRE involves both global and specific regulation. When the ion level is low, ion regulation protein (IRP) bind firmly to the IREs on the mRNA. When the ion level is high, IRP's affinity for the IRE is lower. Ion metabolism disorder treatment strategy. Gene encoding ferritin modified with weaker IREs to increase ion storage and reduce symptoms of insufficiency. Precise alteration of IRE sequences in genes such as ferritin can decrease ion storage capacity, which would lower their binding affinity for IRP. Last but not least is post-translational modifications. PTM is biochemical modifications or changes that take place on protein after being synthesized from mRNA and translated into amino acid sequences. Phosphorylation of BRCA1 is an example of PTM. BRCA1 remains inactive until it is phosphorylated by specific enzyme in response to DNA damage. When BRCA1 is phosphorylated, its ability to function in homologous combination repair and ensuring the accurate repair of double-strand breaks is enhanced. As for the application, the phosphorylation status of BRCA1 can be used as a biomarker to assess the efficiency of DNA repair mechanism and the response to DNA damage. It also aids in identifying individuals at high risk of cancer and helping in early diagnosis.
In conclusion, the regulation of gene expression in human cells is a complex and multifaceted process. It involves key steps such as transcriptional regulation, RNA processing, translational regulation, and post-translational modifications. These steps ensure the accurate and timely production of proteins, which are crucial for cell and organism function. When these processes go awry, they can lead to genetic disorders, cancers, and other diseases. Understanding these mechanisms opens up therapeutic strategies, highlighting the importance of continued research in this field. Next, future research should also focus on several key areas such as deepening the public's understanding, integrative approaches, novel therapeutics, epigenetic regulation, personalized medicine, and environmental factors.
These are the reference for the assignment. That's all for our presentation. Thank you.